Hello everyone, I'm Nagendra Babu, and first of all I would like to thank the organizers for giving me the opportunity to present my work here. And I'm going to talk about how talent-mediated gene editing was used to study the distribution and dynamics of IP3 receptors and the calcium signals they evoke. To start with, I'm going to give a brief introduction. IP3 receptors are calcium release channels in the membranes of endoplasmic reticulum, and they release calcium upon binding IP3 as well as calcium itself. And they are the essential link between several extracellular stimuli that activate the G protein coupled receptors that release IP3 and a variety of calcium regulated cellular processes. The calcium signals released from IP3 receptors can be local, such as those released from a single channel, and the calcium released from a single channel can go on to activate a neighboring channel by a mechanism known as calcium-induced calcium release. And this allows regenerative propagation of calcium signals into hierarchy of calcium release events, thereby a local calcium signal can become a more global calcium signal. And it is the spatially and temporally organized calcium signals that allow calcium to selectively regulate a variety of cellular processes, including gene transcription, contraction, and exocytosis. And the underlying spatial architecture of the IP3 receptors in the endoplasmic reticulum membrane is very important because it determines the propagation of these calcium signals and thereby controls the cellular response. Calcium signals from the native IP3 receptors have been visualized, but without any information from the architecture of these IP3 receptors, because there was no opportunity to look at them in live cells at native levels of expression. Immunofluorescence, you can look at the distribution, but you can't see the calcium signals because they are fixed. Overexpression, and that perturbs this spatial arrangement. So there is a genuine need to look at the native IP3 receptors for their distribution and dynamics and to study calcium signals simultaneously. In order to do that, we use gene editing. I'm not going to go too much in detail into that. Briefly, I used talents to tag endogenous type 1 IP3 receptors with EGFP in HeLa cells and isolated those cells by fluorescence activated cell sorting. And these cells, the tagging was accurate in these cells. As we can see, there is a band corresponding to the EGFP-tagged IP3 R1, and there were no off-target tagging as suggested by this in-gel fluorescence, which allows visualization of the EGFP-R1 protein directly in the lysates. So there is only one band. That means that nothing else is tagged. It's only the the IP3 R1 is tagged, and they formed functional channels. Then I used total internal reflection fluorescence microscopy to study how they look, their distribution, and their dynamics. They formed puncta. Most were mobile, some were largely immobile for protracted periods. So I asked these following questions. What do these puncta represent? Why some are mobile, some are immobile? And where are the calcium signals in relation to their mobility? To answer the first question, in turf images, EGFP-IP3R1 localized with a marker of ER, confirming that they are expressed in the endoplasmic reticulum. And within ER, they formed discrete puncta. To quantify the number of IP3 receptors within this puncta, I used stepwise photobleaching, whereby the stepwise reduction in EGFP fluorescence is used to compute the number of fluorophores within one puncta and thereby the number of IP3 receptors within them. This revealed that an average punctum had 2 IP3 receptors within it with a range of 1 to 13 channels. This suggests that these are clustered channels. These results were also confirmed by the super resolution imaging, and here there is a turf image of a single punctum that's magnified for clarity, and here is a stome image, and the yellow dots are the approximate size of an IP3 receptor that you would expect. Within some of the puncta, IP3 receptors were tightly packed, and within some other puncta, IP3 receptors were more loosely packed. This diffuse distribution, sometimes the distances between two IP3 receptor channels is too great for them to be interacting directly. That suggested that there may be some scaffolds in place that holds these IP3 receptors together, either in an immobile site or in a mobile site, that allows them to move together as a single cohort. So in conclusion, endogenous IP3 receptors are cluster channels. Next, I studied their mobility. Most of the IP3 receptors were mobile within the ER, and I used single particle tracking to quantify their mobility within the ER. And here are the single particle trajectories color coded according to their track. If IP3 receptors movement within the ER is purely by random diffusion, then the relationship between mean square displacement and time should be linear. But what we find is a superlinear curve. This suggests that there is some directional mobility. Actually, some puncta are moving directionally. Then I analyzed these individual single particle trajectories, which reveal that there are 4 distinct kinds of mobility within these IP3 receptors. There is a immobile population, and there is a large diffusive population, and some that go by confined motion, and there is a clear population that moves by directional motion. Further experiments revealed that dynein and kinesin motors, which are molecular motors on the microtubules, contribute to this movement. In the interest of time, I'm not going to go into the data. If someone is interested, I can talk about it later. So in conclusion, IP3 receptors are mobile by both diffusive and directional motion, and kinesins and dyneins contribute to this directional movement. Next, I looked at the calcium signals. And first, it is important to establish what is a mobile site and what is an immobile IP3 receptor. So by overlaying pseudo-colored EGFP-IP R1 images separated by 30 seconds, immobile IP3 receptors were uniquely identified as white spots. So whatever that is white here is an immobile IP3 receptor because it hasn't moved for 30 seconds. And then I used a red calcium indicator, Cal590, to study the calcium signals after histamine treatment. So histamine evokes these discrete calcium release puff events in the cell, and these calcium release puff events co-localized with immobile IP3 receptors. And for clarity, I have just highlighted one of these spots here. There's a calcium release event, an immobile IP3 receptor, and here is the co-localization. And this is also confirmed by a plot profile along this line. It is the immobile IP3 receptor, and that's where calcium release happens. IP3 that is necessary for the activation of these IP3 receptors can be delivered by global means, which is by photo-uncaging caged IP3, which is more uniform delivery, and as well as by receptor activation such as histamine. Both reveal that it's the immobile IP3 receptors that initiate calcium signals, that the calcium signals almost exclusively initiate out of the immobile IP3 receptors. So I conclude that it is the immobile IP3 receptors that are the calcium release sites. Calcium release from these IP3 receptors causes activation of a calcium entry into the cell to refill these depleted ER stores. And the STIM, which is a ER calcium sensor that regulates this store operated calcium entry, accumulates at sites near this immobile IP3 receptor. These white spots and the red spots are the STIM. So this suggests that immobile IP3 receptors are optimally placed at the ER-PM junctions, ER plasma membrane junctions, whereby they can receive the IP3 generated from plasma membrane to release calcium. At the same time, they can also regulate the calcium entry pathways into the cell to allow store refilling. So in summary, IP3 receptors are cluster channels, and they move by diffusion and directional motion. Kinesin and dynein motors contribute to their directional mobility along microtubules, and it is the immobile IP3 receptors at ER-PM junctions that initiate calcium signals. And that would be all. A quick acknowledgements to the lab, my supervisor, Professor Colin Taylor, and to the funding bodies, Wellcome Trust, BBSRC, and Cambridge Trust. And thank you all for listening.